at VIT.
Okay. Yeah.
Yes, that's right.
Yeah.
The conference that we met.
Yeah. Almost 2 years, almost a little longer than 2 years. Yeah.
Yes. Not yet. We are about to drive with Dr. Bhatt's leadership. We are about to drive. Some more students are the true beneficiaries of listening to us, no?
Yeah.
Doctors are doing a phenomenal role in terms of getting these seminars going and so we'd like to. Where are you now at present?
I'm actually in Vadodara. I mean, because I'm originally from Vadodara, I grew up here.
Okay.
And my parents still live here. So usually between Ahmedabad and Vadodara, because grandparents live in Ahmedabad and parents live here. So kind of between two places.
If you're in some part of Karnataka, I thought we'll ask you to introduce us to a few places to join the IM Bangalore chapter. So
yeah, no, I normally I would travel to like VIT for sure, but this year, with all the things going on, just staying with my parents, which is also good because I'm getting good time to stay with my parents at home.
Right, absolutely.
So.
Vashek is also BBB, born and brought up in Baroda.
Oh, okay.
Okay.
Her father was in Sarabhai Chemicals.
Okay. Which is no more anymore. Sarabhai is all gone and they have like
sold off their properties.
Sold off big apartments and shopping malls and everything. So you still have family in Baroda? You still
no,
have
we
family?
have sold our all property now. So we have like my brother is also there in Bangalore. So we have all settled over here only.
Okay.
Basically, we are from West Bengal. Because of my father's service, I born and brought up in this one.
Oh, okay. So you probably speak also Gujarati.
Yeah, I can speak. But in Baroda, generally you don't require because it's very cosmopolitan.
Very cosmopolitan. Yeah.
You can understand everything. Not in.
Yeah.
It was a very nice. Actually, it's a very nice place to stay.
Yeah. I prefer it over Ahmedabad because Ahmedabad is too crazy and too much traffic, too many people.
You have good facilities, medical, you say, education, you say.
Yeah.
It's not that crowded like Bangalore or any other cities.
Yeah.
Also, they miss too much.
Yeah.
maybe knowing your brother Gautam.
Yeah, he passed out he was maybe 5 years, he's elder to me, so he has passed out in electronics engineering from MS University only.
Okay.
He was just slightly maybe 2-3 years maybe senior to you.
Yeah.
Yeah, share screen.
Should I try to share it or maybe later?
Sir, you can also do that. But you can just try once, sir, before everybody gets admitted. You can just try once.
Yeah, let me try. So in that way, so it's going to stop your screen and then it will start.
No problem, sir. No problem. You can try once.
Okay. Let's see.
Yeah.
Are you able to see my screen?
I'm able to see.
Yeah. Perfect.
Okay.
Perfect, sir.
Okay.
Perfect.
Great. It's always nice when technology works.
Good.
Maybe for I said I had to struggle actually. Because
it's just so strange, right? Sometimes the technology will cooperate, and in last 7-8 months, we have all struggled with trying to do screen shares. So
and that to MS Team, Zoom.
So many.
Yeah, our university is using Teams, so we use Teams, but it's all the same at the end.
Yeah. Yes, sir.
Excited over it. Camera off. Why on? We'll wait a couple of minutes more.
Yeah. Still people are joining, sir. Okay.
Yeah, people are joining.
Yes, sir.
Yep. Sure.
Since most of them are at home or a cup of tea, so it's not a question of office time. Probably Vaishak is still there in the office. Because we start our day at 7 o'clock, 7 to 3. Yeah.
Okay.
Camera on, is it?
Yeah, good evening all. Welcome to you. We will be just starting this program in another couple of minutes. Yeah, I think Dr. Bhatt, sir, you can start welcoming.
Shall I start?
Yeah, you can, sir. You can.
Some more people will be joining? Okay, let them join.
Let them join, sir. Let them join.
Yeah. Good evening to you all. Listeners who are joined from India. Anyway, Professor Ketul C. Popat is also in India. Good evening to you, sir. On behalf of Indian Institute of Metals Bangalore chapter, it gives me immense pleasure to welcome you all amidst us for the monthly webinar. We would like to continue this series of holding the regular webinars in the chapter. Probably this is third in the series. And I must acknowledge the effort of Dr. Deepa in arranging this lecture. And I also thank Professor Ketul Popat for readily agreeing to deliver this talk. Beginning February 2021, Professor Amol Gokhale, President IIM, requested all the chapters to hold series of webinars, workshops, and conferences to commemorate the Platinum Jubilee year of the Institute, that is University of Metals. I have tentatively prepared a schedule for the first 6 months with the help of Dr. Deepa and others. I will discuss this in the meeting, coming EC meeting, to how to go about, and I request all the members to give their input and also contribute in this mission of IIM head office. So we have planned to hold a series of lectures, at least one per month. I think the Dr. Krishnan from Bal Subramaniam from DMRL is the coordinator for this, identified by Professor Amal Gokhale. Professor Amal Gokhale is currently the president of IIM. Most of you know or no, I don't know. He was ex-DMRL director. Now he is with the University of Technology at Bombay. So he's professor over there. So he's now, for this year, will be the president of Indian Institute of Metals. So he has requested all of us. In this regard, Professor Chandan Srivastava had a 3 days students related seminar. So, and we'll continue this. We have to give our plans how to go about. So I once again welcome all those who have remotely joined for this seminar. May I now request Vaishakhi, the Joint Secretary of Indian Institute of Metals Bangalore chapter, to introduce the speaker to the listeners. Vaishakhi, please.
Good evening, one and all. It is my immense pleasure to introduce our today's speaker, Dr. Ketul C. Popat, who is professor in Department of Mechanical Engineering, School of Biomedical Engineering, School of Advanced Materials Discovery at Colorado State University. He is Associate Department Head of Mechanical Engineering and Director of Undergraduate Programs in Biomedical Engineering. Prior to that, he was working as a research specialist in Department of Physiology at University of California, San Francisco. He has authored over 130 peer-reviewed publications in journals such as Langmuir, Biomaterials, Journal of Orthopedic Research, Journal of Biomedical Materials Research, etc., and has H-index of 41. He has also presented his work in numerous national and international seminars and conferences. He received his PhD in bioengineering from University of Illinois at Chicago in 2003, MS in chemical engineering from the Illinois Institute of Technology, Chicago in 2000, and BE in chemical engineering from Maharaja Sayajirao University, Baroda in India in 1998. With this brief introduction, I will invite Dr. Ketul C. Popat for his virtual technical talk on micro and nano engineering of material surfaces for tissue engineering and regenerative medicine. Please, sir, over to you.
Thank you. Thank you for the introduction, and thank you everyone for coming for this presentation. I will share my screen, and hopefully everything will go fine, because it did go fine just a minute back. So can everyone see my screen?
I can see.
Yes, we can see, sir.
Okay, great. So so in the next maybe 40 minutes or so, I will give a brief sort of overview of some of the research that we do in my lab, specifically on material surfaces, how we can engineer material surfaces, specifically at micro and nano scale, so that these material surfaces can be a part of a medical device. And these medical devices, when they're used in the body, you don't have any sort of rejection that would happen from the tissue perspective. I have been at Colorado State University for about 13 years now. I started in 2007, almost 13 and a half years, 2007. And ever since I've been there, my lab has been focused in this area. My lab is also called Biomaterials Micro Nano Engineering Lab. Apart from being a professor and doing all the teaching, as in the introduction it was mentioned, I'm also the associate department head in mechanical engineering and director for undergraduate studies in the School of Biomedical Engineering as well. I'm going to try to give a very brief overview because, with the time constraint that we have, I'm going to sort of give a brief overview of some of the work that we specifically do on metals or alloys in my lab. And I always, before I start my talk, I always say that I'm not a material scientist. I'm actually a materials engineer. In my lab, we don't make new materials. We don't synthesize new materials. We take existing materials and we try to modify these materials to see how they would perform in a specific type of a biological environment. Before I actually begin, I just wanted to kind of have some acknowledgements that without these funding agencies, I don't think I will be able to give the presentation. My lab has been funded over the past number of years, from National Science Foundation, National Institutes of Health. These are sort of the main funding agencies that fund all our work. But apart from that, there has been small funding from the state, some non-profit organizations. I haven't listed some industrial funding here, but we do have some industrial funding as well to sort of evaluate basically, material properties or engineer material surfaces for various different type of applications. I also have to acknowledge my students. I think through this pandemic times, I have gone in my lab maybe 3 or 4 times in last 7-8 months. My students have been there pretty much every day since May. And so, I really have to thank them because it's their hard work, which is the reason why I can actually come in front of all of you and give the talk on, the work that they are actually doing. So my lab has a few PhD students, few master's students, and some undergraduate students. Normally, I do have a lot of visiting students who come from India, from Brazil, from other parts of the world. Because of pandemic, I just have one person from Brazil who actually came in January and was stuck. I think he will go back again this January, but we had drastic reduction in terms of visiting students, visiting researchers from different parts of the world that normally would come to my lab and work on different aspects. I should also acknowledge my collaborators. I have actively been working with many people across US. I have listed some of them here, but I've also worked quite a bit with people across different countries, specifically from Brazil, obviously from India, and Canada, South Africa. These are some of the collaborations that I have very actively been working on. Even through the pandemic, we have still continued working on these collaborative projects. And I feel like this is very important, especially the field that I'm in. It's important for us to have this international collaborations because we're trying to solve problems which affect all across the world, people all across the world. Especially medical devices is an area which is not very local, it's a very global field. And so we do need a team of global engineers to solve this problem. Just to give a brief background about where I come from, the university is in the state of Colorado, and it's in the city which is called Fort Collins. The city of Fort Collins is actually at the foothills of the Rocky Mountains. I just put a map here so that for some of you, if you're not sure, you can kind of get an idea geographically where the university is. This is Denver, which is one of the big metro cities. This is typically where you have the international airport, and we are about 70-80 miles north from Denver. And we are actually right at the foothills of Rocky Mountains, but we are a very small town actually. So our population is 150,000. 1.5 lakh doesn't seem too much when we compare to places in India, but we are the third largest city in our state. So we are considered as the third largest city. It's a very small place, but just to kind of give you some pictures, it's a very old place. It was one of the places where people from East used to come and kind of rest before they go into the Rockies to do all the exploration for the gold, which they never found. So during the gold rush time, it was a very popular place because it was right at the foothills of the mountain. So this is typically what we see from our university. We have Rocky Mountains, which right now they're all actually covered with snow, but in the summertime, it can be pretty nice. The university is also very old. It was formed in 1870, predominantly as an agricultural university because that was the main focus. Then it became agricultural and mechanical because moving parts were really important to have good agricultural production. And that's how kind of the university got started. But over the time, the university has become very much involved in research areas in many different fields. And right now, our university is very well known for veterinary research. They're very well known for atmospheric research. They're very well known for basic science. Basically, chemistry is very well known. Apart from engineering, which is a very growing field, there are many different areas which are very well known. But we have about 27,000 students, about 1,600 faculty members. So we are not a very large university, but we are still a moderately sized university. Predominantly, we cater towards the people who live in the state of Colorado. So a lot of our students are local students. A lot of our students are first generation students, which means their parents have never gone to university. We have a lot of Hispanic student, which is basically the people from people who speak Spanish language. And so we are very diverse in that way. Just to give you an idea about the university. So, I just have some pretty pictures of the university. This is how it looks like in summer. This is one of the areas. This is how it looks like in winter with a lot of snow. You can imagine that there is a drastic difference. And this is how the same area would look like if it was an autumn. So we do have very distinct 3 seasons where we have summer. And, normally I would not be in US in the summer. I'm normally in India in the summer, so I'm very used to the heat. But this was the first time after many years I had to stay in US for the summer because you couldn't travel anywhere. And summers are pretty hot in Colorado as well. Our temperature can go all the way up to 35 degrees C, and it can remain like that for at least a month. So we do have quite a bit of summer as well. This is just to give you an idea about the university. This is one of the areas where, normally when there are students on campus, this is like the central hub where everything is happening. This is our student center. We have a stadium, where the team plays football. My lab is specifically located in this building, which it's a relatively new building. About 6 years back, we built the building. And my lab is a pretty typical lab where we do a lot of material stuff. So you have fume hoods and things like that. And we also actually have like cell culture facilities because we do a lot of work with stem cells and things like that. So I hope this kind of gives everyone a very brief overview of where I come from and the university. In my lab, we focus on biomaterials, and we basically focus on surface of biomaterials. And as we know, materials have been used for medical applications for many years. I mean, if you think about dental implants, they existed thousands of years back as well. Now, in those days, they didn't know that gold could be used as a material, but it was still used. They didn't know the science behind why it works and what are the properties, but it worked. And so we know that materials can be used for different type of medical devices. These days, if you look at medical devices, about 10 to 12% of the world population has medical device. And these medical devices can range anywhere from a simple device like a contact lens that you wear on the top of, the topical contact lenses, or it could go really complicated as things like stents or hip implants. But if you look at it, we use metals, we use polymers, we use ceramics, we use composites, we use all sorts of classes of materials for different types of medical devices. As a matter of fact, some of the newer medical devices which are coming, they're also living products. Basically, these are the ones where you also have cells which are part of this medical device. So it becomes a living product, and the material kind of provides a template for these cells to actually grow and live. And when that implant is put, when the device is put, those cells help in the regeneration process. So we have come quite far along in terms of how we use materials for different types of medical device applications. What we focus on is on the surface. And the surface is a very important part of a material because that's the first thing that comes in contact when a material comes in contact with the body. When a medical device is placed in, the surface is sort of the predominant interaction. Depending on the properties of the surface, you could have protein adsorption, then have cells, you could then have eventual tissue formation and things like that. But if your surface does not have good properties, if it doesn't have appropriate properties, then you may end up with sort of rejection process or immune response that can happen. And your material would basically, or the medical device would basically be useless if you have this kind of interaction that happens. So from an engineering perspective, we have a lot of things we can do to the surface. We can engineer the surface, not just the chemistry of the surface, but also the topography of the surface. And by doing so, we are now either creating more biomimetic surfaces, which are very much like how the biochemical structures are inside, or the topographies are the kind of structures which are actually found inside the body. And so that's what the focus has been in my lab. And we kind of start from very basic. When we think about how we want to engineer surfaces, we kind of go back to like, what is that surface intended for? What is that medical device intended for? That medical device is intended to come in contact with the tissue. And when it comes in contact with the tissue, there are cells in the tissue, and those cells are actually going to see the material surface. So we want to make sure that the material surface properties are such that these cells are actually able to migrate to the surface, grow the new tissue, and eventually you have this, this integration process that's going to happen. So we need to understand biology. And that's one thing I tell my students, because I have students who come from material science, who come from mechanical, who come from biomedical. Biology is sort of the core. We don't need to be experts on biology, but we need to understand from an engineering perspective, what does biology, what is in, what we need to know from that biology. So when you think about a tissue, from a very engineering perspective, we can classify tissue as cells and everything which is not the cells. Everything which is not the cells is called the extracellular matrix. And cells are living in the extracellular matrix. Cells are actually producing extracellular matrix also, but the extracellular matrix provides a support mechanism that the cells need. And combine that with a bunch of other things is what our tissues are. But if you start looking at sort of the interactions which the cells are facing and the extracellular matrix is made up of, cells, they interact with each other using, proteins. They interact with each other using extensions that come out. There's a lot of interaction that happens, which is sort of governed by the biochemistry around those cells. But that biochemistry is actually provided by the extracellular matrix, which has proteins, which has small molecules, and all of these have a unique size. They do have a chemistry, but they do have a size. As a matter of fact, when you look at tissues, just to give you a simple example, we have a protein, we have a molecule called collagen, which is in our body. There are many different types of collagens, but chemically it's the same. Structurally, it's very different. But the same collagen is in the bone, and the same collagen is in the skin. It's a different type, but chemically it's the same. Skin is not stiff like bone. And so there is a lot of structural aspect which is involved, which sort of goes back to the basics of materials. And we can use some of those principles to understand how tissues inside the body and mechanics of tissues inside we need to put it on our surface so that when that surface comes in contact with the tissue, you have proper integration and there is no rejection that will happen. Now I'm going to talk about two areas mostly. And again, as I said, I'm not going into a lot of depth in these areas just so that I can cover a few things that I want to do. The first thing to talk about is orthopedic implants. And specifically, I want to talk about load-bearing orthopedic implants, such as knee implants and hip implants. Traditionally, these are made from titanium and titanium alloys. Basically you can see this is a hip implant picture here. You have this metallic stem that actually goes into the marrow cavity. And, for many years, cemented implants were used where you put a cement that actually binds the bone with the material. But over time, there have been issues with cemented implants because there are cracks which are formed. There is debris that can form. The debris can also form because you have the ball and the cup joint where, the inner lining of the ball is high density polyethylene. And sometimes wear is going to happen and those wear particles can get stuck into the gap. A lot of people have this kind of implants and a lot of people have a lot of problem with these kind of implants. So these implants actually fail. And then every 10 to 15 years, about 7 to 10% of people have to get a revision surgery. Revision means you're removing the implant and you're putting back a new one. So imagine the first surgery is really traumatic. The second one would be really, really traumatic. And so the idea is that how we can create materials so that these material surfaces are such that you have a good integration, you don't have any failure of that interface that happens. And so the field has moved quite a bit from cemented implants to press fit implants, but you can't just make a smooth surface. You can't make a smooth surface because if you make a smooth surface, it's not going to work. You have to have some level of complexity on the surface that matches the complexity of the tissue. Now, I'm just going to very quickly go through some of the slides. I'm sure everyone knows about titanium and alloys. Titanium is, pretty versatile material for a lot of orthopedic applications because it has good corrosion properties, high specific strength. As a matter of fact, many different implants are made from titanium-based materials. This is just a cartoon which kind of shows that, you could have hip implants, pins, knee implants, dental implants, stents. There's so many things which involve titanium-based materials. And existing implants, which are there in the market, which are made from titanium-based materials, do have some sort of a surface structure. Because you can make, as I said, you can make smooth implants. There is some level of roughness which is there, and different ways in which these roughnesses are created. A lot of these techniques, they use plasma-based processes. So you use titanium plasma spray coating, and you make the surface very rough. Typically about few hundred microns is what the roughness of the surface is. And what that roughness does is that now when that implant comes in contact with the tissue, the cells will migrate, go into that rougher structure, grow the new bone, and then eventually bond. We still have problems with that. We still have failure that actually happens. And so a lot of the new methods are using something called hydroxyapatite. Hydroxyapatite is the mineral content of the bone. So it's really not, it could be any type of calcium phosphate that could be used as well. So you can put calcium phosphate on these implants, and then you can texturize the calcium phosphate. So now you are giving a physical topographical cue, which is the roughness, but you're also giving a biochemical cue, which is the calcium phosphate or hydroxyapatite, which is there. But none of the commercial products do have a nanoscale topography. We do need a nanoscale topography, and I'll just talk about that, very briefly here. That if you look at the bone, it's a major part of our tissue. It's a major part of our bone. It's a very big tissue, and if you start dissecting bone into its components, what you find out is that bone is actually made up of nanomaterials. Hydroxyapatite crystals are nanoscale. Collagen fibers are actually nanofibers. They are arranged in a certain way, so you can have nanopores, micropores. So it's a very complex structure. And right now we replicate microscale features on implants, but we don't do nanoscale. So the idea is that maybe we still do a microscale structure, but on top of that microscale structure, we have some type of a nanotopography that matches the nano size scale, which is actually present in the native tissue. So the idea would be that you take hip implant, for example, you provide this nanotopography in certain regions. You don't need to do it in the whole implant. You only need it into certain regions where you need those anchor points for the tissue to actually grow and form these bonds. And so that's what we have been focused on. We have been mostly focused on how we can take existing titanium alloys and how we can actually provide different kinds of structure. One of the surfaces that we have worked quite a bit is titanium nanotube arrays. This is a very simple process. It's a very simple electrochemical anodization process where titanium is anode, and we use platinum as a cathode. We put it in an electrolytic cell, and we use an electrolyte, a fluorine-based electrolyte. So we use hydrofluoric acid, which is a pretty dangerous acid, but if you follow all the precautions, then it's fine to work with. And just by controlling the voltage, just by controlling the time of reaction, by controlling the concentration of the electrolyte, we can have very unique nanotopography. And we are not depositing anything. We are actually doing a bottom-up process where we are taking the bulk material and transforming into these nanotubular structures. So just to give you an idea, if you change the voltage, you can change the diameter. And so we have some control over that. If you change the electrolyte, you can change the spacing between. So you can have these tubes touching each other or these tubes far apart. So there's a lot of control in terms of what we can actually do in terms of giving the different kind of topographies. And we have done a lot of work taking these surfaces and trying to evaluate how stem cells interact. And we find that diameter affects the spacing effects, the wall thickness of these nanotubes also actually affects the cellular response. And so we need to find a sweet spot where we would have the optimum performance of the cells. At the same time, the surface should be such that we should be able to create on an actual implant. It shouldn't be that difficult that we can't use implants commercially available and actually do that. The interesting thing about creating these nanotubular surfaces is that these surfaces are extremely hydrophilic. Contact angles less than 10, so they are super hydrophilic, which kind of helps for cell migration as well, because cells like to migrate and form connections. So the better surface wettability, the better it is for the cells as well. And we do a lot of work with contact angle, and trying to understand sort of the theory behind why it is the way it is. We do a lot of work with stem cells, as I mentioned. We have done a lot of work with mesenchymal stem cells. These are the stem cells which are from bone marrow, and we normally do that. I'm actually showing here some work that we have done with adipose-derived stem cells. Adipose-derived stem cells are actually present in the fat tissue, and a lot of fat tissue is actually discarded these days with surgeries, breast removal after women go through cancer, or liposuctions, which are actually done. So there's a lot of fat tissue that gets discarded, which is rich of these adipose-derived stem cells. So we can remove these stem cells. They're very much like bone marrow stem cells. And in a way, this is a more exciting approach, because you don't have to go in the bone marrow and get the cells. You can actually just do fat biopsies and get stem cells. And so we thought, let's try to use these cells and see if these surfaces are actually able to make bone cells on them. And so we have done quite a bit of work with this adipose-derived stem cells, where we take these stem cells, we put these stem cells on these surfaces, and we allow the cells to grow. And the idea is that we want to know if the nanotube surfaces support a better growth of the cells. And we do find that, as a matter of fact, because they're very hydrophilic, we do see that in 7 days, when we start with very few cells, we can end up with a whole surface that's covered with cells. This is what we would want when we want the integration of the implant to happen. We want the cells from the natural tissue to go to the implant surface, attach, and divide. And division is not just the only thing that we want. We also want the cells to start producing bone, which is sort of the second phase of the study that we do. But just before I go that, we do a lot of electron microscopy to see morphology of the cells. The previous figure that I actually showed you was a fluorescence, which tells us about the cell structure and cell functionality, if they were alive when they were stained. Whereas electron microscopy purely would tell us how they're interacting with the surface. How does a nano surface interact? And as you can see with some of these images here, the cells are much longer. They have much longer extensions if they are actually on nanotubes compared to that on titanium. Then we do differentiation. This is something where we want the cells to actually form bone. And so we go through this whole series of process where we provide cues to the cells, and the cells would then become bone cells, because we want them to become bone cells and produce proteins and things like that of bone. And so, we do quite a bit of work in this also. So here is an example where, we have cells which are covered with, the surface is completely covered with the cells. But because now they're becoming osteoblasts, they're producing a protein which is called osteocalcin. And we can see green dots here. So by the way, the red is the cytoskeleton, blue is the nucleus, and the green that you see here is all the protein that the cells are producing. And we find that the cells do produce a lot more protein on this nanotube compared to, not that titanium is bad. Titanium is still fine. It still works. It still, has good properties, but we want to make it better. And that's why we use, we look at this Osteocalcin expression. We do a lot of other work in this bone area. I'm just kind of giving a flavor of what we do. We have done a lot of work with modifying chemistries and not just topographies. We have also done quite a bit of work with putting these implants into animals and trying to see that how the tissue is growing around them, and do pull-out tests and things like that to see the strength of the new tissue that's formed. And so we continue, this is an ongoing project that we have in the lab, and I have students who continue working on that. The other thing I want to talk about is cardiovascular disease. And as a matter of fact, this is the leading cause of death in the world, and it costs a lot. And when I say cardiovascular disease, it's a disease related to blood, or it's a disease which is related to blood vessels, or disease related to the heart. As you can see, this is the map of United States, and I'm sure there are maps like this for India as well, where you can see the areas where the density of cardiovascular cardiovascular disease is much higher. And as a matter of fact, well, 2020 is very unusual. 2020 had more deaths because of COVID-19 than any other disease by now. But normally, cardiovascular disease is killing a lot of people. And so, we need to figure out ways in which we can prevent this disease from happening. But if the disease has happened, how do we mitigate that disease? And how do we basically, cure
disease, which is specifically related to um, blood vessels, is basically narrowing of the blood vessels, and that happens because of deposition of things like plaque and fat and calcium and things like that. And so normally when the blood is flowing, it's flowing at a normal pressure, normal flow rate. But if you have a plaque, that's actually going to now change how the flow is happening. Brain and heart and kidneys and legs, and so there's a lot of different things in the body that would actually get affected. So the solution
to this is basically to do a percutaneous coronary intervention. This is typically where angioplasty is done. And a small mesh-like tube is placed in, which is basically a stent, so a stent procedure. The other procedure is where you literally go into a bypass, and that's a bigger procedure because you have to stop the heart and the lungs and connect the person to a heart-lung machine, and then use polymer grafts to replace the damaged part. I won't talk about the polymer part. We worked quite a bit on this as well. I'm mostly going to talk about the stent part and how we can engineer stent surfaces to prevent some of the negative effects that happen with the material. So this is typically what happens. You put a wire mesh. This is typically made from nitinol. It's made from nitinol because it's a shape memory, so you could do minimal invasive surgeries. A lot of these surgeries are now a day surgery. You go in the morning, and if everything is fine, you can come back in the evening or maybe the next day. These don't require patients to stay in the hospitals for a really long time. There are two types of stents which are there. You have the bare metal stent and you have the drug-eluting stent. Bare metal stent doesn't have any coating. Drug-eluting stents, they actually do have a coating. And I'll just talk in a minute what is the difference between both of those. So the bare metal stent doesn't have any coating, and when that's actually put inside the body, it would actually, open the blood vessels, so you could maintain the flow. But within few weeks or month, if this metal material, if the metal is coming in contact with the muscle layer, so we have endothelial layer, we have muscle layer, and we have the fibroblastic layer. If it comes in contact with this muscle layer, the muscle tissue gets very irritated in the blood vessel and the cells start growing. And they would start growing this tissue like this. So it would start pushing the stent. So the whole reason you put the stent to keep the blood vessel open, the stent material is causing that. To resolve this, drug-eluting stents came. So they have a drug that's going to stop the division of the cells. So it does actually prevent this restenosis condition. But after all the drug is finished, you have thrombosis that happens where the blood clotting happens. And this is also very dangerous because once you have blood clot inside the body, and if the blood clot is released and if it's free flowing, aneurysms can happen, it can go in the brain, it can be very dangerous. So there are two issues, the restenosis issue, the thrombosis issue. In my lab, we focus on both, but I'm going to mostly talk about the restenosis issue. How does the material surface come into play here? The material surface is really important because the process of this thrombosis, which is the blood clotting, starts with protein adsorption. You have proteins that actually get adsorbed, and that allows the platelets in the blood to attach, and these platelets would get activated, and then the red blood cells get trapped, and there's a bunch of different things happen which results into blood clot. There are other things that can happen as well, where you can have rejection of the implant itself. Restenosis can also happen, but I'm mostly going to focus on this thrombosis part today. But we do work on each of these different issues that could happen with material surfaces. So we use titanium nanotube arrays as as interfaces for these stents. I just wanted to check, I still have a little bit of time, right? Maybe 10 minutes?
Yeah, we have time. So
okay.
still, we have
Okay.
15-20 minutes.
Okay, great. Because I didn't want to rush too much, and I cannot see the time when I'm sharing the screen.
15-20 minutes.
Okay. So we use titanium nanotube arrays as interfaces. And so the idea is that you take stents and you grow these nanotubes on it, which provides an architecture. And I'll talk about in a minute why this architecture we think would be a good architecture to have. But then these nanotubes are also delivery vehicles. So you could actually put drugs inside. The drug-eluting stents have a coating, and that coating has a problem because coating will wear off eventually. And then you can have coating particles which would be floating in the blood. Here, we are not using a coating. It's basically coming out from a delivery vesicle. And the delivery vesicle itself is the one that's providing the topography that is actually needed. So the idea is that we do this. And the reason why we think this would work, because this structure of nanotube very much actually matches the structure of the blood vessel, inside of the blood vessel. So if you take a cross section of the inside of blood vessel, you find that, so this is a blood vessel, you find that there are these hair-like structures which are there. These are the structures which are produced by endothelial cells. And this structure is called endothelial glycocalyx. These hair-like structures are on the surface of the blood vessel, and they are the best known surface that's going to prevent all the negative interaction, because for a healthy individual, blood is flowing. It's only when there's a damage to this layer, the blood starts clotting and everything happens. So we know that this is a very good anti-clotting surface, a very good surface that prevents restenosis. So the idea is that can we match the surface like this? So you have a stent that goes inside the blood vessel, but the inside of the stent, if we create these kind of nanotubes which match this endothelial glycocalyx, and when the blood is flowing, the cells, the proteins, they're still seeing that nanotopography. And maybe that would help us in improving the properties of the stent surface. So we have evaluated the surfaces by themselves before modifying them. So we do actually use blood. We use human blood, and from the human blood we remove the platelet-rich plasma. So you take the blood and you put it in centrifuge and you spin it at a certain speed for a certain amount of time. All the clear things will come on the top. The red blood cells will settle down. The clear things are where you have all the proteins, you have the platelets and the white blood cells. We use that, and then we put them on our material surfaces. And so you can see here, the green images are the fluorescence images, which basically shows all the live cells. So you can see on a titanium surface, which doesn't have any nanotopography, a lot of clusters are seen. Whereas on nanotubes, we see very sparse cells. We still see cells, but very sparse. In SEM, you can kind of see, so the big ones, the small ones are all the platelets. The big one that you see here, that's the white blood cell. And you can see here that you have a white blood cell interaction happening with platelets, whereas here we do see white blood cells, but we don't see these kind of clusters which are formed. This is very interesting behavior because that's what happens in the blood clotting process inside the body, where platelets and white blood cells interact with each other and do certain functions that goes to the next step of the clotting that happens. So we do know that topography does affect. We do know that topography is making a difference in terms of how the cells interact with the surface. We also wanted to see how blood clotting happens. And so we do a very simple assay. We take a drop of blood, we put it on a material surface, and we allow the blood to clot. And we observe that blood clotting that's actually going to happen. And we find out that even the rate at which the blood clots and also the blood clotting is lower on these nanosurfaces, partly because of the surface area effect and partly because of the topography that is more biomimetic for these cells. This is still not enough because we still have clotting which is there. By the way, before I go to the approaches, I also wanted to see that we also looked at some immune response, because that is also important, because we know that stents don't elicit immune response in terms of the rejection, only has problem with restenosis and blood clotting, but we could have immune rejection with these nanostructure, which means basically complete rejection of the implant. So we have done that as well, where we have taken the white blood cells from the blood and we have put the white blood cells on these material surfaces. Again, the green is showing the whole cell. The blue is actually showing the nucleus. Green means that the cells are alive. And you can see here that we do see less cells on titanium nanotubes, but we don't see these foreign body giant cells that you actually see here. So these foreign body giant cells are the ones which actually would go to the further process of rejection. So we want to avoid these foreign body giant cells, and we have been able to achieve that with these nano kind of surfaces. But as I mentioned, this is not enough. We still want to make these surfaces more better. And so now we have relied quite a bit on modifying the chemistry of the surface. One of the chemistries that we have worked quite a bit is this polyelectrolyte multilayers. Basically, these are polycation, polyanion, layer by structures. And by doing so, by controlling the number of layers, we can control the thickness. And we can also mimic sort of the chemistry of the blood vessel surface. So we have the topography of nanotube that the material gives, but we don't have the chemistry. So can we also have the same chemistry that's actually present on the blood vessels? So we have used 2 polycations, 2 polyanions. We have used chitosan, 10-flock, heparin, and hyaluronan. Heparin, hyaluronan are very well known, have been very commonly used. Heparin has been very commonly used. 10-flock is a new, sort of a newer material. And what we found out was that 10-flock worked really well. And this is something that's derived from tannin, from the leather processing. So it's still biologically derived. And chitosan, I think it's a very common, polymer that has been used. So we have used, 2 polycations, 2 polyanions, and we have built this layer by layer structure. And the first thing that we wanted to see is, can we maintain the topography of the surface? So we did 5 layers and 19 layers, and you can see 19 layers, the whole surface is covered, which means we lose the topography, which we don't want. So we went ahead with this 5 layers, where we can still see the nanotubes, and we don't see this complete covering of the surface that actually happens. So we went with this 5 layers, and then we did a lot of studies with this 5 layers. So the first thing that we wanted to do again is the same thing. We wanted to see how the cells will interact. So we again took the blood, remove the platelet-rich plasma, which has platelets, proteins, and white blood cells. We put these on the material surfaces for a certain period of time and see what happens. And what we found out was that the 10-flock heparin combo had by far the least. We still did it on the 5 and 19 layers. 19 was okay. We didn't really have the topography here, but it was still okay. But after this, we decided we'll just go with the 5 layers because we were actually able to see quite a bit of reduction in the number of platelets which are there. Nanotube by itself, as you can see, is very much. Now, when you see these images, they look very different from, nanotube here is very different than nanotube before, because there's a lot of donor to donor variability. We take the blood from a donor, so somebody may have more cells, somebody may have less cells, but we normally look for the trends. We look for the percentage changes, we look for the trends as to how much reduction is happening, because people to people, there's going to be a lot of variation in the blood. The other approach we have used is nitric oxide chemistry. Nitric oxide is a potent vasodilator, and it's known to inhibit platelet activation. As a matter of fact, nitric oxide is released from our blood vessel wall at very small amounts in our blood, and it helps in this process. And so basically, we took these nanotubes, we built these polyelectrolyte multilayers. We used here chitosan and heparin, and then we put these nitric oxide releasing moieties inside. So as these layers are degrading, and again, these layers are very small. They are less than, less than 10 nanometers, less than 5 nanometers as a matter of fact. So we could release this nitric oxide. Again, we want to make sure the chemistry is not changed. So the chemistry does not change the topography. So we still see that we can maintain nanotubes, and then we can see the release. The release is within minutes. And it's really hard to work with nitric oxide being such a small molecule. So we are now working to try to push these release limits to days and then eventually weeks by changing the chemistry and by protecting those nitric oxide molecules. And we also want to have sort of a zero order release. We don't want a burst effect. We want a sort of a zero order release where continuously that nitric oxide is coming. But with the way we had nitric oxide right now, we still could see a significant difference in terms of the platelet adhesion and activation. So here are some images of titanium. You can see a lot of platelets. Nanotube still has some. Nanotubes which had this nitric oxide, we could barely see any platelets. So in a way, we are pushing towards creating a surface which would not have any blood clotting that would happen on the surface. Now, the other issue is that it's not just that we don't want blood clotting, we also want the cells to attach. So that's the next goal, and I don't have results on that yet. So we want to create something that doesn't clot the blood, but allows the cells that we want to attach. They should be able to attach and they should be able to grow. We have also done a lot of work with superhydrophobic surfaces. That is the other approach that we use. These are non-stick surfaces. Lotus leaf is a very good example of a superhydrophobic surface. And the reason why it's a superhydrophobic surface is because of the chemistry. It has a wax kind of a coating on it, but also there is a nanotopography that's actually present. And so our idea was that can we use the topographies that we make, combine it with something like a carbon fluorine bond, not really Teflon, but use a carbon fluorine bond. And then can we then create these superhydrophobic surfaces on which we have practically no attachment or no adhesion of proteins that would happen. And so there's a lot of theory behind it. We would prefer something to be in a Cassie-Baxter state. So we create a topography. And when you create a topography and when you control the gap between the topography, you can trap air in between. And when you put a liquid droplet, it would never touch the surface. The air pressure is going to the pressure is going to be exerted and this droplet would remain as a droplet. Now, one of the questions that obviously comes is that if the air is trapped and if the implant is inside the body, what's going to happen? Well, we have done a lot of studies now in the past few months, which shows that for a prolonged period of time, the air remains trapped. And there have been some modeling work which has been done, which also shows that this air is trapped for indefinite period of time. So we should be able to maintain this superhydrophobicity. And so that's what our goal is. And this is a surface on which we made superhydrophobic layer. And you can see the blood drop remains a drop, plasma remains plasma, and the water, we added blue dye so that we can see it's a water. It remains as a drop. It's not spreading. And if it doesn't spread, if it spreads, then you have attachment. If it doesn't spread, then you probably you won't have that attachment. So we have made different types of these superhydrophobic surfaces by using different types of silanes, a chlorosilane, a fluorosilane. And I'll show in a minute that the fluorosilane was actually much better than the chlorosilane because the carbon fluorine bond, we know from experience with Teflon that it creates, hydrophobicity. But the contact angles of these surfaces were above 150, which is where we are in the superhydrophobic regime. Still, we were not actually able to change the topography, which is good because we can still maintain them to look like nanotubes. I have a few videos here, which basically are going to kind of show a little bit about how the droplet interacts. So here is a titanium surface, plain, it's clean, but no modification on that. And you can see the drop of the water, what happens. This is what we expect to happen. If you modify this titanium with fluorosilane, you see that the surface is hydrophobic, contact angles a little bit more than 90. So this is what we would expect. This is nanotubes, which have that very high surface area. They're super hydrophilic. And let's watch the nanotubes with the fluorosilane. It's a little slow motion because you will see what I'm talking about. The drop of the liquid is just bouncing. So imagine that if blood was flowing, blood components will not even touch the surface. They would just basically slide from the surface. If they don't touch the surface, slide from the surface, there is no chance for the proteins to adsorb. There's no chance for the cells to attach, and we can practically, prevent blood clotting from happening. Just preventing blood clotting is not enough because we want cells to attach. So these kind of surfaces will be truly non-stick. Nothing is going to stick on. Nothing is going to stick on this surface. We have done a lot of work with looking at protein adsorption, looking at factor 12A activation, which is an important sort of a protein in the step process that happens. And we consistently find out that the fluorosilane was much better. So even though the chlorosilane did have superhydrophobicity, it was still not that better. It was the fluorosilane which was actually the best. And then we do a lot of imaging, and you can see here that the fluorosilane still had quite a bit less number of platelets which were actually there, and activation of the platelets was also much lower. Lastly, I have some images which is actually about the blood. So hopefully people won't mind if I'm showing blood in my presentation. But this is kind of showing about the flow part. So we have some videos of the flow, but they're much more complicated to kind of visually see. So this kind of is mimicking what would happen if the blood was flowing. So you can see here that this is plain titanium, a drop of blood, and you can see that the blood is spreading like how you would expect. The previous video was on water, this is on blood. And if you lift this, you can see that the blood is sliding, but it's actually also making a streak, So this is where clotting is going to happen. Let's look at the nanotubes. The nanotubes were the super hydrophilic ones. And when you put a drop of blood on these, you see it spreads. Now, this was better than titanium. If you remember the few slides back when I showed, not the best. It was still better because you could very quickly drain the whole surface and you won't have this streak formation. You could literally drain the surface. You still have some cells which are there. This is the chlorosilane. So you can see the drop of the blood and you can basically see that the drop remains a drop. It's not spreading like the ones which were here. And if you lift this a little bit, you would see that the drops are just going to slide. The student is a little bit nervous to take this video, so it's a little slow than the other ones. But you can see the surface is practically clean. The last one is that fluorosilane, and you can see here the drop of the blood is remaining the drop, and it doesn't even want to stay on the surface. So even without lifting the surface, the surface has so low surface energy that it just doesn't want to stay on the surface. It just slides out. So this doesn't really mimic the flow setup, but it kind of gives an idea that, how the blood would interact. And as I said, we are doing a lot of flow studies more recently to show that superhydrophobicity is affecting, but also superhydrophobicity is being maintained because we don't want superhydrophobicity to go away. Otherwise, the stents will be the same as drug-eluting stents. Well, all the drug is gone and now we have the problems which are happening. So we want to maintain the superhydrophobicity for a substantial period of time. We do a lot of work with whole blood clotting as well, as I mentioned before. This kind of looks at how the blood is clotting. So the more changes you see, that means more blood is clotting. And we found out that with the chlorosilane, there's no changes which are happening. This is basically measuring the hemoglobin, which is in the blood. So you put a drop of blood and the blood is clotting, which means the hemoglobin is getting trapped on the surface, but the unclotted blood still has free hemoglobin. So this is measuring that unclotted blood, unclotted blood hemoglobin. So those are the results that I have. We are continually working on, on this. And as I said, my students have been working even in the pandemic with social distancing, with shifts, because the lab that we have, no more than 2 people are allowed. So they are trying to work at odd hours, at weekends. So, hats off to them for working really hard. And sometimes I feel like I'm not doing much because I have not gone to the lab and I'm just staying at home and mostly spending my time on, Zoom and Teams meeting. But they have worked really hard, and I think they definitely deserve the credit for all the things that I'm talking here today. So just to sort of briefly conclude or summarize, so nanostructured surfaces do affect how the cells respond. Now, the fact is that they're not going to be favorable for all the cells. I didn't talk about some of the results where nanostructured surfaces were not favorable, but it's not the answer to a solution. Nanotechnology is not about an answer to a solution. It's just an approach which allows us to think about things a little bit differently. It also opens up a lot of different ways in which we could analyze materials and understand material properties. But in our case, nanostructured surface for orthopedics did actually make a huge difference. Nanostructured surfaces made a slight difference for blood, but as soon as we modified those surfaces with polyelectrolyte multilayers or put nitric oxide or had superhydrophobic surfaces, we had huge advantages. And so with this, I would conclude my talk. We sort of work in the field of nanotechnologies, not so much in terms of, as I said, more engineering, taking materials and engineering and using nano approaches, but our focus is also on biological interactions. So we sit sort of in the intersection of these three different sort of areas where we work with materials, we use nano approaches, but we look at biological interaction and we try to understand how these interactions happen, and in turn, can we learn something so that we can improve the material surfaces even better, so we end up having less problems with implant rejection inside the body. With this, thank you very much for listening to my talk. I'm very happy to answer any questions if there are any. If there is not enough time to ask questions for many people, or if you end up having questions later on, feel free to send me an email as well. I am still here in India for a few more weeks, so I We'll be happy to talk on the phone as well. Being on the same time zone, we don't have to worry about me sleeping and people being awake here, or people sleeping here and me being awake there. So it's a little bit easier. Thank you. Thank you very much.
Yeah. Thank you, Professor Ketul Popat, for your quite an informative talk. Yeah, you have taken us to a different world from the conventional material science and metallurgy presentation to an interaction of material science and biology. As a chemical engineer, it's really amazing to hear your diverse area of activities, wherein a combination of metals and alloys, material science and biology, how to show how materials can be engineered to suit to human body. And just before this, my wife was mentioning that you also mentioned in a presentation that biology should be there as a part of their curriculum. I think Manipal Institute of Technology, MIT, has realized that, and it seems in their first year, instead of technical English, during our days, it used to be there as technical English one paper. Now, instead, they have introduced biology for all the engineering stream. Probably it's quite useful. Really, in that way, your presentation was good. And my son also had to study that one paper. He's in the 4th year now. In the first year, he has studied biology as one of his papers. With this, I request participants to ask any questions if they have any.
Yeah, anything is there, I think they can ask. Otherwise, I think we can.
I think.
Yeah. Yeah, Mr. Shashwat, you raised your hand. Can you please unmute your audio and can speak?
Yeah, thank you, sir. So, very insightful talk. One question I had is when you talked about the polyelectrolyte to mimic the chemistry of blood vessels. So, what's the effect of number of layers there?
So the effect of number of layers, so the reason why we looked at the layers was because we were also thinking at the same time to put nitric oxide. So to be able to release more nitric oxide, we thought we'll put more layers. But obviously more layers from a surface perspective, it's not going to make a lot of difference because from the biochemistry would be still the same. But we lose that nanotopography because it completely covers it. And so that we don't want it. So we were trying more layers so that we could put more nitric oxide, which I didn't show with more layers here. We ended up doing 5 layers only. But from the chemistry perspective, what the cells would see, the chemistry would be the same. Doesn't matter if it's 5 layers or 19 layers.
Okay, and one more thing I had is like in the end when you talked about superhydrophobic materials, you mentioned that they are also not the ideal ones as they would not allow any cells to attach. So then what's the motive here? Like what cells would?
So see, ideally, when we think about stents and even the current stents, they don't do that as a great job. So, you put a stent, a wire mesh, that stent is already touching, the outside of the stent is already touching the blood vessel. And the blood vessel layer is the endothelial cell layer. It's a very thin layer, and underneath you have the smooth muscle cell layer. We do not want that endothelial cell layer to get damaged. If it gets damaged and if the smooth muscle cell layer is exposed, then we end up having restenosis. So the goal is to not damage that endothelial cell layer, but rather encourage those endothelial cell layers to grow so that they can cover the whole surface of the stent. And after a few months, when the blood is flowing, it won't even see the stent. It will only see the new endothelial cells, which is the same as the old and the same kind of problem. That we cannot do with superhydrophobic. Now, we can still do it where outside we don't put superhydrophobic coating, but inside we put superhydrophobic coating because inside we don't want anything to attach. But the outside, we want that integration to attach. And so when I mentioned that cells would be harder is, we want true integration. That process is called re-endothelialization. So we do want re-endothelialization while preventing blood clotting, which I think with superhydrophobic would be a little bit difficult to achieve because superhydrophobic means nothing is going to attach.
Yeah, thank you very much.
Yes, Dr. Bhatt, sir, you can unmute and can speak.
Any other questions, or if there are no other questions, I request Ravi Kumar of Trinity NDT to propose the vote of thanks for Atul Professor Ketul Popat's presentation, Professor's presentation.
Yeah, thank you. Good evening all once again. I would like to express my sincere thanks on behalf of the Indian Institute of Metals, Bangalore chapter, for presenting an excellent talk, which is very novel and which is very closely connected with metallurgy, material science, nanotechnology, and even a lot of things about biology. And it's really amazingly brilliant session, what we feel, and we would like to thank you, sir, for taking your valuable time and then presenting yourself this valuable information to all the members of Indian Institute of Metals. And I would like to thank Dr. Deepa Srinivasan, our Chief Engineer, Pratt & Whitney, for coordinating this session with you along with others. And I also would like to thank Dr. R.R. Bhatt, who is also currently the chairman for Indian Institute of Metals, Bangalore chapter, for organizing this program and coordinating with you and completely bringing in this quality program with all the members. And I would like to express our sincere thanks to the Honorary Secretary, Bangalore chapter, Indian Institute of Metals, treasurer and various office bearers and executive committee members of Indian Institute of Metals, and also all the members of Indian Institute of Metals who are present over here. And apart from that, various other societies like Indian Institute of Welding and a lot of other society members also are present over here. And I would like to thank all the participants who have participated in this program through Zoom, and also we have made a live session on Facebook as well, and a lot of people have watched from there also. And I would like to thank all of you for being over here, and thank you all, sir, once again for presenting at this session. Thank you.
Thank you very much, and thank you for inviting.
And one more, Mrs. Vaishaki, nicely she has introduced the speaker. I would like to express sincere thanks on behalf of Indian Institute of Metals. Thank you so much.
Thank you very much.
We have a photo session.
I request all the participants just to have a memory. We would like to, have your video switched on. Please unmute your video so that we can have one snap with all of you, which can be memorable. Right? Please switch on your video if it is convenient to all of you. Please, I request all of you, please, wherever possible, please unmute. Yes, good. Yes, others? Anybody? Within another 10 seconds, if you can switch on, we will.
Am I visible?
Yeah. Yes, completely, sir. You are visible.
Thank you.
Yeah. So I'm taking the snapshot, just another 10 seconds, another 2-3 members are there. Yeah. I think others are not comfortable. Mr. Shivashankar Reddy and Radha Krishna, Nitin Narayan, and Hemant. If you're not comfortable, I will take the snapshot. Otherwise, yeah. Sure. So I request all of you to smile, and I will take one snapshot. Yeah. Sorry, sir. Give a smile. Just 1 second. 1, 2, 3. One more. Just 1 second. 1, 2, 3. Just 1 second. Thank you all once again for being over here. Particularly special thanks to Dr. Ketul Popat, also our chairman, and Deepa Srinivasan. Thank you all. The session is adjourned.
Yeah. Hope to see you again. Yeah.
Thank you, sir. Thank you so much.
Bye.